Good afternoon. Welcome to Genius Week and today's webinar, Metagenomics as a Window into the Microbial World, Medical Implications of the Gut Microbiome and Other Applications. I am Dr. Damian Urena, Business Marketing Leader for Next Generation Sequencing at GeniusWith. I am pleased to introduce our featured speaker for today's session, Dr. Gilad Kedem, Senior Bioinformatics Analyst at GeniusWith. Dr. Kedem pursued graduate studies in biomedical engineering at Johns Hopkins University and obtained his doctorate in medicine degree from Sackler School of Medicine at Tel Aviv University. Prior to joining GeniusWith, Dr. Kedem was Chief Molecular Officer and Chief Technology Officer at a Metagenomics-based startup engaged in infectious disease diagnostics. Please note, if you have any questions for Dr. Kedem, kindly use the chat room to submit them, and he will address them at the end of the session. Without further ado, Dr. Kedem, welcome.
Thank you, Damien, for this introduction. I'm excited about the opportunity to talk with you today about the burgeoning field of metagenomics. In the limited time we have, I can only give you some highlights, but hopefully it's enough to give you a sense of the field and to pique your interest to learn more. I'll talk initially about an introduction, and then technology, applications, and then we'll conclude with questions and answers. So what is metagenomics? Metagenomics is a study of the totality of DNA recovered directly from any location. The goal is to characterize the communities of microorganisms present in these locations and to understand how they influence their surroundings. The term metagenome was coined in 1998 by Dr. Joe Handelsman as, quote, the collective genomes of soil microflora. And the field has really exploded since that time. Metagenomics can be used to study any microbial community, which is also called a microbiome. As you can see in the figure, microbiomes reside in soil, human gut, oceans, and other sites. The influence of microbiomes across any of these diverse ecosystems is enormous. So it's very important to understand microbiomes. Metagenomics is used to study any unknown mixture of nucleic acid sequences. Since all microbes and viruses contain either DNA or RNA, metagenomics is a window into the full microbial world. It sheds light on all three domains of the tree of life: bacteria, archaea, and eukaryotes, as well as viruses. Metagenomics can also be used to analyze larger eukaryotes, whether it's dinosaurs or even human beings, for forensic purposes. What is so powerful about metagenomics? So metagenomics constitutes the most precise tool to look at microbial communities in their specific natural locations. And just to give it some context, in the 17th century, the father of microbiology, Leeuwenhoek, invented the microscope, making us aware of the existence of microbes. In the 19th century was developed the laboratory cultivation techniques for cell bacterial culture. In the second half of the 20th century, there was developed molecular biology and genomics, and now we have metagenomics. And metagenomics is the only tool to study microbial communities in their totality. Metagenomics is a very hot field. If you look at the PubMed term metagenomic, you can see that since the word was coined in 1998, it's been increasing exponentially. Some of the highlights of in the short history of the field is the Human Microbiome Project in 2008, the firm Second Genome, one of the earliest microbiome firms, was founded in 2009. In 2015, various major universities launched microbiome initiatives, whether it's the Center for Microbiome Informatics and Therapeutics at MIT or at other places. Finally, just last month, the White House launched the National Microbiome Initiative, which we'll talk more about soon. What are the types of analysis or goals we have in metagenomics? The first primary analysis is simply what microbes are present in the sample. To do that, you have to first subtract any host DNA. Next, you classify each sequence read into its taxonomy node. Then you estimate abundance, and you can also do microbial assembly, genome assembly. Next, you can ask, what are the microbes doing? And to do that, you can use metagenomics to look at functional genetic potential, and then you can use metatranscriptomics to study actual functional profile. You can also do comparative analysis, where you can compare diversity within a sample, called alpha diversity, diversity between samples, called beta diversity, and also gamma diversity, which is over a larger region. There are two main types of metagenomics. One is targeted genomics, metagenomics, also called amplicon sequencing. It's the most common method. It's less expensive, but the taxonomic resolution is only to the genus level in most cases. To do targeted metagenomics, you have to pick a genomic marker and then develop primers to create amplicons for that region. The genomic marker should be common to all organisms of interest, but also display enough heterogeneity to be informative. So we typically choose the ribosomal genetic locus. For bacteria and archaea, typically we use the 16S rRNA, which is a small subunit of ribosomal RNA. And for fungi, we use the small subunit, the large subunit, or the internal transcribed spacer regions. Shotgun metagenomics is much more powerful, but also more complicated. It directly examines all DNA in the sample, not just target genes. So it can identify all microbes in a sample, whether bacteria, archaea, viruses, fungi, whereas targeted can only really identify those for which you create the right primers for. Shotgun metagenomics lets you go down to the species and subspecies level in resolution, and also enables functional analysis. So in metagenomics, as for all NGS workflows, it starts off with a project design, then you move on to nucleic acid extraction, library preparation, sequencing, and then you move on to data analysis and interpretation, which is the bioinformatics part. It's important to realize that in the wet lab and also in the sequencing, parameters and choices made there can have a big influence on the final bioinformatics results. So when you do the sequencing, you have to ensure that you don't introduce too much or any amplification bias. And also, when you do the sequencing, you have to have high enough quality so you don't create chimeras when you do assembly, or also have high enough depth so you can detect rare microbes in your sample. There are many bioinformatics categories for metagenomics, and new software in the open source world is being developed all the time. Every few months, there's another important package. So you really have to keep up with the bioinformatics and choose the best-in-class software for each class. For targeted metagenomics, like shotgun metagenomics, it starts off with quality control, programs like FastQC and Informatic, and then the software splits off in specific software for targeted metagenomics and for shotgun metagenomics. We also have functional analysis software, whether it's to examine signaling pathways or metabolic pathways, to study protein families, protein-protein interaction, and so on. It's just a small sample of bioinformatics software. For targeted metagenomics, CHIME, Quantitative Insights into Microbial Ecology, is a great open source framework that wraps popular tools. It uses the Green Genes 16S database. Regarding shotgun metagenomic sequence classifiers, which is a class of software, in 2007, Megan was developed, which uses BLAST against reference databases. Later, Thimble was developed, which also combines it with Markov models. And in 2014, Kraken was developed, which uses k-mers, or exact subsets of fixed length, or subsegments of fixed length of a read. And that system, k-mer, is very fast with high sensitivity and specificity. Visualization is very important in metagenomics. One example is Krona, which is a very intuitive and exploratory tool to look at taxonomy. Some of the major projects in metagenomics have been the Human Microbiome Project in 2008. Later, at the same time, we had MetaHit, which is the metagenomics of the human intestinal tract. This was a European project to classify human beings into 3 enterotypes, which is similar to genotypes or blood types, but it looked at gut microbial composition. And finally, the National Microbiome Initiative, announced in May 2016, is a public-private partnership for studying microbiomes across many ecosystems. And there are many federal agencies involved, as well as over 100 external institutions, including the Gates Foundation, the Juvenile Diabetes Research Foundation, and companies like One Codex. There are many types of applications of metagenomics. I'll start off with the medical ones. So you can use it for infectious disease diagnostics. You can also use it for microbiome analysis, whether for the gut microbiome, for irritable bowel disease or diabetes, for microbiome-based diagnostics, and finally for microbiome therapeutics. Other applications in the medical realm are for outbreak mapping, for epidemiology, nutrition guidance, and studying the characteristics such as virulence factors of the pathogens. So using metagenomics for infectious disease diagnostics is ideal for polymicrobial infections, where you need to know the mix of pathogens involved and not just the single pathogen involved. For example, in diabetic foot ulcers. It's also important for critical disease, where we have to have the diagnostic performed quickly, like in sepsis. And finally, when other diagnostic techniques fail. Advantages of metagenomics versus bacterial culture include that metagenomics is comprehensive, whereas culture cannot cultivate most species. Metagenomics is unbiased, so the physician doesn't have to form an a priori hypothesis regarding the pathogen, whereas culture depends on selecting the appropriate growth media. Metagenomics is much more accurate, going to the subspecies level, and it's also much faster in that it can perform under 24 hours as opposed to bacterial culture, which can take several days. One of the most interesting uses, or maybe the earliest use of metagenomics for infectious disease diagnostics, was a case of the 14-year-old boy who traveled to Puerto Rico. This boy had a type of immunodeficiency, and after swimming in a river in Puerto Rico, he developed brain inflammation called encephalitis. So encephalitis is actually caused by over 100 types of pathogens, and neither culture nor other serological tests could determine the causative agent causing the encephalitis. So the boy's life was at stake, and UCSF was able to identify the causative agent as Leptospira, which is a difficult to culture organism, using their, at the time, a research-based software system. And once the correct organism was identified, the boy's life was saved with the right antibiotic treatment. So there are several, now there are several types or examples of metagenomics pathogen detection software, including SERPI from UCSF and Pathoscope from Boston University. And there are several startups in this space, including PathoQuest in Paris and Cosmos ID in Maryland. Another major application of metagenomics in medicine is to study the human microbiome, which is the sum of all bacteria in and on the body. It's considered a second genome, and it's very much influenced or involved in health and disease. There are up to 10 times the number of human cells, of bacterial cells, than there are human cells. And there are 100 times the number of human genes in the microbiome. And unlike the human genome, the microbiome is dynamic, so the genes and gene products and pathways are constantly in flux. There are actually many or several local microbiomes in the human body. The gut microbiome, which weighs up to 2 kilograms, may be thought of as a separate organ. It has a major role in immune modulation, metabolism, and gut-brain axis. And dysbiosis, or an unhealthy imbalance in the microbiome of the gut microbiome, causes several classes of disease, including autoimmune diseases like type 1 diabetes, metabolic disorders like type 2 diabetes, and CNS and other neurological disorders like multiple sclerosis and even autism. Other human microbiomes include the respiratory microbiome, which has a big effect on asthma and cystic fibrosis and other diseases. The skin microbiome, which influences atopic dermatitis. And then the vaginal microbiome, which plays a role in bacterial vaginosis and even preterm labor. And the influence of the microbiome on new diseases is a hotly researched area, and new associations are being discovered all the time. Microbiome-based therapeutics are a very interesting area of metagenomics. The most basic is fecal microbiotic transplantation, where stool from a healthy donor is placed rectally in the colon of a sick patient. And this technique, while perhaps somewhat unappealing, is extremely effective for treating recurrent Clostridium difficile infections when other modalities or like antibiotics don't work. The next refinement of this concept is to mimic FMT through pills taken orally, and Series Therapeutics in Cambridge, Massachusetts, has the most advanced microbiome candidate for C. diff infections. Other approaches include designing rationally bacterial cocktails, or even engineering, genetically engineering bacteria. One company, Evelo Biosciences, also in Cambridge, is developing an oncobiotic platform to disrupt tumors, cancer tumors, by interfering with metabolism and interaction between the tumor and the surrounding human cells. Finally, there's small molecule drugs, which are being developed by Enterome and other companies to modify how bacteria interact with the gut. Another big application of metagenomics is for agriculture. One example is the rhizosphere, which is the thin soil region surrounding the root of a plant. It's extremely dense in terms of bacteria, And the microbiome and plant interact very much together. One influences the other. The plant secretes things like proteins, sugars, and other things that influence the microbiota. And in turn, the bacteria secrete products like signaling molecules and antibiotics, which influence the plant. Enrichment of plant microbiomes, in other words, enriching the plant microbiome with the right bacteria, can lead to enhancement of plant immunity, control of plant pests, increase plant growth, and also enhance crop fitness, for example, for drug resistance. Other applications of metagenomics are for the environment, for example, to study water quality or climate change, and industrial applications include food chain safety and manufacturing. So in summary, you can see that metagenomics is a revolutionary and exciting technology which can study microbial communities as they exist in many ecosystems. The challenges are numerous because metagenomics is a very new field, and there's still not many standards. For example, most microbial species have not yet been sequenced, so reference sequence databases are still insufficient. Few standards exist to validate microbiome measurements or workflows, and there are few systems-level bioinformatics platforms. Finally, it's even unclear what constitutes a healthy microbiome, whether in human, gut, soil, or the environment. As far as future directions, it's difficult to know how the field will evolve, but one area that interests me is combining multi-level omic data and other data to produce a holistic picture of a human being or a patient. And that data includes genomics, transcriptomics, and proteomics, as well as the patient's electronic health records, which contain medical treatments and outcomes, and also to include within this profile metagenomic data, metatranscriptomic data, and metaproteomics. So that's the conclusion of the talk, and I'll be happy to answer any questions if you have any. For listening to the talk, my name is Gilad Kedem, and I work at GeneWiz in New Jersey.
Thank you, Dr. Kedem, for your insights. We will now open it up for questions. Again, please enter your questions to the chat room, and we will address as many questions as possible. Okay, so we have a question coming in here for Gilad. One of your slides mentioned nutrition guidance as an application of metagenomics. Can you elaborate more on this topic?
Versatile dietary guidelines, such as the FDA food pyramid, have limited utility. For example, an article from 2015 in the journal Cell looked at how diet affects blood glucose levels and showed that there's a high variability to people's response to identical meals. The paper examined a group of people over 1 week and monitored their blood glucose levels and meals. Specifically, looked at postprandial glycemic response, which is a risk factor for type 2 diabetes, cardiovascular disease, and other diseases. In addition to finding that there's a high variability in response to identical meals, the authors created a machine learning algorithm that predicts glycemic response based on the gut microbiome and other clinical parameters. And the study showed that a tailored diet guideline based on this algorithm lowered glycemic response, and as well as altered patients' microbiota. So I think that in the future there may well be personalized nutrition guidance based on the gut microbiome.
Thank you, Dr. Kettle. We have a couple more questions here. One of them is, does the shotgun approach offer expression analysis capabilities?
So metagenomics, as we know, is a powerful way to look at abundant levels of microbes and gene content and potential function of the microbiome, but not at the actual gene activity. And metatranscriptomics shotgun sequencing allows whole genome profiling of gene expression. Typically, in metatranscriptomics, as far as the wet lab goes, you isolate total RNA from the microbiome, enriched RNA, and then after fragmentation, you convert the RNA to cDNA with reverse transcriptase. You construct the library, and then the sequencing is performed. As far as the bioinformatics, there are really two approaches. The first is that you want to map the RNA sequences to reference genomes and pathways. For example, you can map metatranscriptomic sequences to the KEGG database, the Kyoto Encyclopedia of Gene Genomes, and then obtain actual pathways of expressed genes and see how these pathways are up or down regulated depending on different conditions like health and disease. The second approach is to perform de novo assembly of the new transcriptome, and the idea is to obtain full transcripts as well as gene isoforms for different species.
Okay, thank you. We have time for one more question, and the question is as follows. Are there efforts to synthetically engineer microbiomes to improve crops quality or human health?
Yes. So there are several ways to engineer microbiomes to have a positive effect on their host. One approach is to use synthetic biology to artificially engineer bacteria and then insert them into the microbial mix. Another approach is to engineer microbiomes by allowing the host, whether the host is a plant or animal, to influence the microbiota over several generations, a process called host control. Through host microbiome interaction, whether involving, for example, resistance or immunity, the host recruits beneficial bacteria into symbiosis and then excludes other microbes. So the idea is to select hosts with positive traits, for example, plants that have drug resistance or rapid growth, and then allow the host to shape its own microbiome. And I think the host microbiome selection, what's interesting about that is it allows microbiome engineering without any knowledge of the actual microbiome composition.
Okay, thank you very much. I think actually we have one more question that just came in. We'll ask this last one. Do you think it is possible to do reliable target metagenomics with MiSeq sequencer?
So yes, you can use the Illumina MiSeq machine to do both targeted metagenomics as well as shotgun metagenomics. And the nice thing about the MiSeq is you can do fairly long reads. You can do 2 by 250 base pair reads, paired reads, and it's proven to be a very effective system for both 16F and shotgun.
Okay, thank you very much, Dr. Kedem. I also want to thank everyone attending this webinar. This concludes our session.